Okay, good morning, everybody. It's a great pleasure for me to be here, and I'd like to thank very much Giulio and his team for organizing such an amazing symposium. I must admit it's really challenging because there is an ambitious proposal here that try to bring stimulation from micro scale up to the patient scale, actually, not only major scale or macro scale. And so my task here is try to provide some information on the recent state of the art of literature in which non-invasive brain stimulation is actually emerging as a new tool that comes from the neurophysiology ground, but then can be transferred into the clinical ground for its unique properties. That it's basically to test the principal aspects of pathophysiological changes occurring in Alzheimer's disease patients. And so I will start with obviously what we know so far. It comes from studies that use the different methods, especially transcranial magnetic stimulation and obviously transcranial electrical stimulation, that can be used to probe the activity of certain areas of the brain in several neurological disorders. And especially I will refer to the recent findings in the field of Alzheimer's disease and dementia. So this is very simple simplification of the state of the art. So what can we do here? We can use actually non-invasive brain stimulation like RTMS or TESS to do two things basically that can be important in the clinical context. One thing is actually we might think that if we standardize this procedure and we actually understand which part of the brain we can stimulate and which is the readout that we want to obtain, we might end up with some biomarker of synaptic dysfunction, which is clearly important due to the feasibility of non-invasive brain stimulation. We can speak about this later. On the other end, we have the obvious implication, which is you may actually understand better which network are dysfunctional, which part of the activity of the brain needs to be repaired, let's say, and then this could be translated into novel therapeutic approach. So in my talk, I will try briefly to underline the recent aspects of these two potential implications. So let me start with the history of TMS in Alzheimer's disease. So it is kind of surprising because now we have sort of a long history that started almost 15 years ago, in which people started to use TMS of the motor cortex by simply measuring the amplitude of the motor evoked potential. A few things show up in the last 10 or 15 years. To summarize this, what it was constantly found is that if you apply single TMS pulse over the primary motor cortex, what you see in Alzheimer's disease patients is that the threshold is actually decreased as compared to age-matched controls, meaning suggesting a form of hyperexcitability in the primary motor cortex. But then you can use more let's say, relatively complicated protocols using pair pulse TMS, and then you can evaluate a GABAergic activity in these patients, mainly GABA A. But also, if you use this short afferent inhibition protocol, which basically relies on the peripheral stimulation of the medial nerve at the wrist, and then after 20 or 25 milliseconds, you develop a single TMS pulse on the contralateral primary motor cortex. This results in an inhibition of the motor evoked potential, and this very simple phenomenon actually is strongly related to the cholinergic activity. So these different parameters have been constantly replicated in several studies, and actually short afferent inhibition is strongly reduced in patients with Alzheimer's disease. So at least we know that this could represent a marker of dysfunctional cholinergic activity in these patients. So, but the next step now is try to, put this things that are considered to be like isolated in the neurophysiological community into the ground of the clinical application, which means we have to connect this with the more important evidence in the field of Alzheimer's disease with the underlying pathophysiological mechanism. So what we try to do is to combine this neurophysiological evidence with underlying CSF pathology. So for instance, in this paper that we published a few years ago, we actually showed for the first time that there was a robust correlation among cholinergic dysfunction and the underlying levels of CSF A beta, but there was no correlation at all with CSF tau. So again, suggesting that this cholinergic dysfunction likely is the consequence of amyloid beta deposition and not tau neurodegeneration. And what is also important for us, and this was a paper that we published in 2009 in Neuropsychopharmacology, is that you may actually use these methods to probe novel therapeutic approaches, because it's important that you can measure in vivo changes in cortical excitability and cholinergic transmission in this patient, and therefore you may actually directly test whether a certain drug may have an effect on these circuits. And at that time, we were the first to observe that actually if you administer levodopa in these patients, you may actually restore the altered inhibitory transmission that is related to cholinergic transmission, and then therefore we observed that actually there's a strong interplay, I mean, dopamine and cholinergic transmission in this patient. This is just an example. But then, of course, we have another great opportunity, which is try to estimate what happens in terms of cortical plasticity, because we know that in at least in animal models of AD, it is kind of established that LTP in the hippocampus, at least, is considered the neurophysiological basis for learning and memory. And in fact, there is a robust evidence in animal models of the AD that if you verify what happens to, let's say, long-term potentiation in the hippocampus in animal models of AD, treated with soluble dimers, oligomers of beta, you may actually observe a robust dampening of this LTP. And this is kind of constant evidence. And moreover, it was also shown that while LTP was, it is severely dampened, there is also a sort of imbalance with LTD that seems to be increased. And this is important because there is a sort of a, shift in the cycle life of neurons, because LTD is also associated with the promoted apoptosis and cell death, while LTP on the other hand seems to be more associated with preserver function and enriched activity. So but the main observation here, which is obvious, is that so far I've been showing you something that is related to the activity of the primary motor cortex, but the primary motor cortex is not associated with Alzheimer's disease, at least in the early stages. Patients with Alzheimer's disease don't show motor signs or motor deficits until, let's say, they develop the full broad spectrum of the disorder. So why should we focus on the primary motor cortex? The answer was at that time that that was the only thing that we could do, because the technique relies on the primary motor cortex. But if you think about it, it's not a bad idea, because actually you are investigating an area which is not directly involved by mass atrophy. And there are probably an area in which you can observe subtle changes that could represent something that might evolve later on in the course of the disease. So that it could also represent a sort of temporal shift to work on an area that it's not primarily affected by the disorder, but becomes affected later. And this could actually represent an advantage if you see in that perspective. So what we have been doing, and others, is try to better understand what happens in terms of cortical plasticity in patients with Alzheimer's disease by using different protocols. So we have not time to go to this technical detail, but there are protocols that rely more on Hebbian plasticity, such as the pre-associative stimulation protocols. On the other hand, theta-based stimulation protocols that were developed by Inzu Wang some years ago actually resemble more the experiments that have been performed in animal models. And I would like to start from this seminal paper by Fortunato Battaglia and colleagues in New York, published in Biological Psychiatry almost 15 years ago, because for the first time they were able actually to show a robust decrease of pre-associative stimulation, let's say, again, like cortical plasticity in Alzheimer's disease, which is very important. But they were also able to demonstrate that in animal models of Alzheimer's disease, actually this alteration of LTP is not only evident in the hippocampus, as it was demonstrated, but it spreads somewhere. So even if you test it in the prefrontal cortex or in the primary motor cortex, there is consistent impairment of cortical plasticity. Therefore, what this paper suggested is that actually this is a general phenomenon that involves possibly the entire cortex, and we don't have to think about Alzheimer's disease being a pathology that is only related to the memory functions, to the hippocampus, but it's clearly a sort of systematic brain disorder. So we have been pursuing this idea, and we've been trying to investigate more systematically what happens when we apply different protocols of repetitive TMS, and we found that actually LTP was severely dampened independently of the different protocols we were using. But on the other hand, LTP seems to go on the opposite direction, and this was consistent with the experimental data that I showed you before. And then we try to extend this evidence in a larger population to try to better understand the influence of aging, because when you talk about Alzheimer's disease, there is also always this potential confound that what you see is not actually related to the disorder, but more to the aging process. And therefore, we had a chance to investigate this LTP-like cortical plasticity in a large population of Alzheimer's disease, starting from, let's say, 50 years up to 80 years, and compare this to a sample of age-matched age controls. And what we found at that time is that actually, sorry, just to go back, obviously we replicated robust LTP impairment, so there was no LTP in patients with Alzheimer's disease overall. But what is more important is that we also measure short afferent inhibition, which is a cholinergic transmission. So what we found at that time is that cholinergic transmission was actually correlating with the increasing of age. So we were seeing actually an impairment of cholinergic transmission that was higher in patients that were elderly, but also in healthy control. So there was no such a difference, although overall patients with Alzheimer's disease were more impaired in terms of cholinergic transmission. There was a decline in cholinergic transmission that was going in the same direction in patients and in age-matched control. On the other hand, there was a clear separation in terms of long-term cortical plasticity impairment. This long-term cortical plasticity impairment was not associated with aging, and so this means that if you have Alzheimer's disease, even if you are 50 years or 55 years, so early AD, you have at the beginning a clear alteration of cortical plasticity, but not of cholinergic transmission. So for us, this was suggesting that actually cortical plasticity disruption is probably a main feature of cortical alteration, which is related to the pathology more than cholinergic transmission. And then we try to combine this again with CSF data, and we found that actually there was a significant correlation with CSF tau levels. And moreover, we also found an important association at time zero. If you evaluate cortical plasticity in this patient and you measure also CSF values of tau and A beta, you may actually observe that patients that have more impaired plasticity and higher level of tau are those who are going to decline at a faster rate. So this is also important in terms of prediction. And then the other thing that you always want to, in the case of AD, is to take in account APOE genotype, because it plays a very important role. And actually, we know that APOE3 and APOE4 patients have striking differences in terms of clinical presentation, clinical progression, potential response to therapy. So it's really important to take into account this difference. And actually what we found at that time is that there is a strong association among CSF tau and the impairment of LTP, which is evident only in the APOE4 genotype group, but not in APOE3. This means that actually this interplay among neurodegeneration and cortical plasticity is peculiar in patients that have the APOE4 genotype, but not the APOE3 genotype, which is important. And I must say, much more elegant evidence was published at the same year, in the same year by Dave Holtzman group that actually showed the same concept in the context of animal models of Alzheimer's disease, including a relevant role for neuroinflammation. So then if you try to transfer this into the clinical ground, as I mentioned, as a potential biomarker, you start evaluating how much this could help you to predict the disease course of cognitive decline. Actually, you may end up with important results. And in this paper that we published in Journal of Neurology, Neurosurgery and Psychiatry 5 years ago, we actually were noticing that actually LTP-like cortical plasticity measured at baseline was a robust predictor of subsequent cognitive decline, even more than underlying, CSF levels, which are biomarker, which are supposed to be obviously sensible in this direction. And also, and of course, we can also transfer these into the earlier phase of the disease. So we started to evaluate what happens in patients with mild cognitive impairment, and this study was published a couple of years ago in brain stimulation. So again, we found that actually there is an alteration of long-term potentiation in patients with mild cognitive impairment, while short afferent inhibition, cholinergic transmission is not able to distinguish very much among these two groups, neither GABAergic transmission. And then again, when you try to use these measurements as predictor of conversion to dementia, which is very important, you may actually see that patients that at baseline have no problem with LTP, so the LTP is okay. They have a decreased chance to decline and to convert to Alzheimer's disease, while their possibility in 3 years to develop Alzheimer's disease is almost 100% if you have impaired cortical plasticity measured at baseline. So that's some of the story related to what you can detect by simply measuring changes in activity of the primary motor cortex. Again, this could represent the basis for subsequent therapeutic approaches. Just a very brief note about our clinical trial that we designed on the basis of neurophysiology. So we started to investigate whether rotigotine, which is a dopaminergic agonist that is released continuously during 24 hours thanks to a patch, actually may have an impact on cortical plasticity. And we found that not only was restoring cortical plasticity, the administration of rotigotine, but actually there was a boost of cortical plasticity. So on the basis of the evidence, we hypothesized that drugs acting of dopaminergic transmission, which is very important for plasticity, and it was mentioned in the model. Actually, we hypothesized that if you enhance this transmission from the ventral tegmental area up to the prefrontal cortex, you might actually see an improvement of dopaminergic function. And for the first, I want to mention this because for the first time, then we introduced the potential of using TMS in combination with EEG to measure actual changes in cortical activity of the dorsolateral prefrontal cortex. And we designed a proper clinical trial that was supported by the Alzheimer Drug Discovery Foundation, a phase 2 clinical trial that led to some positive results in terms of increasing frontal lobe cortical function as measured by the frontal assessment battery, and also the reduction of autonomies of daily living, which is important in patients with Alzheimer's disease. But again, what we were able to observe for the first time in the context of a clinical trial that issues TMS EEG, you may actually see robust changes in activity of the target area. In this case was the prefrontal cortex. So before and after there was an increase of cortical evocative potential, an increase of high frequency oscillation, not only in the alpha band, but also in the low gamma band, which was evident only in the patients treated with rotigidine, but not in the control placebo group. As you can see, after 6 months of therapy, there is a decline of activity, which is not present in the other group.